Good evening. Okay. See. Okay, so let's begin with today's question answer session. Hello everyone, welcome back to the question answer session of the NPTEL course Fundamentals of Protein Chemistry, which is coordinated by Professor Das Gupta from the Department of Chemistry, IIT Kharagpur, and I am Bias Mukherjee from the Department of Bioscience and Biotechnology, IIT Kharagpur. And today in this session, we will talk about protein isolation and purification. Okay, so we will look at the different methods for the production of recombinant proteins, because a large amount of protein is possible to be isolated from recombinant sources, okay, compared to the natural sources. So yes, recombinant production of proteins, and then we will also talk about the different biophysical and biochemical techniques to separate out proteins and get the desired protein, right? And we will also solve questions related to these. Okay, so let's begin. And if the network isn't very good, you tell me, you stop me in the middle and you tell me that you're not able to view the slides or you're not able to listen to me. Okay, because it's raining very much, very heavily here. So there is a high possibility that the connection might just go out in the middle. Okay, so we'll start, and I will not present this in the form of slideshow, because many times you all say that you all are not able to follow up and there's a delay in the slides, right, changing and all. So we'll just go about this way. Right, so let's start with the first slide. Right, so what is exactly, so the topic for today is protein isolation and purification, right? So the first question that will come to your mind is, why do we even need pure proteins, right? So pure proteins are very important because they have applications in many disciplines, and that includes your biotechnology, which can be food biotechnology, it can be enzyme biotechnology, it can be so many different fields, right? Recombinant DNA technology, And then there is medicine, drug design, vaccine development, proteomic studies. There can be so many diverse fields in which you require pure proteins, right? So what is meant by purification and isolation of proteins? Isolation and separation of protein basically refers to the fact that in nature, if you look at this figure on the left, you see that is a natural source of protein. Okay, so in nature, proteins are not present as isolated units. They are always mixed with something. There is fat and there are multiple types of proteins mixed together in the same protein source, right? So there is always a mixture of a lot of different things together. Now we want this particular protein to be separated and in large quantities. So one of the disadvantages of isolating from natural sources is that the concentration of the protein, the pure protein that you get finally, is very less. So to make sure that we get protein in huge amount, we actually prepare this protein in a different host cell. Let's say it's a bacteria, it can be an yeast cell. So these are all recombinant protein production in which we get pure proteins in huge amount. So we'll study about all of these today. It's a very interesting presentation today. So I'll recommend you to go, listen to it and then try to solve the questions, because all of these biophysical techniques, they are very important for all your competitive exams. So make sure you pay attention today. Okay. So in case you're not able to attend the class, you know very well that you are going to get the PPT and the video recording uploaded on YouTube. So you can always go back and check. Okay. Right. So see, proteins can be isolated from natural sources. However, to maximize the yield, they are recombinantly grown in host organisms, typically bacteria, yeast, and insect cells. Right. Plant cells, mammalian cells, using genetic engineering techniques. Then recombinant DNA technology involves the insertion of genes. So let's say that you require a particular protein to be overexpressed and purified. So you take the gene, the DNA sequence that codes for that particular protein, and you chop it off from the naturally occurring DNA. You can even chemically synthesize it, right? And then you can also clone it. It will be present in a vector. So from there you take it out and you place it into the desired vector, and it can be an expression vector, right? And then what do is you insert that into the host organism. So this is culture to produce proteins in large scale, and biophysical techniques are employed in its extraction and purification. Okay, so now we'll go into the details of protein isolation and purification. So if you see this slide, you understand how hectic the procedure of isolating proteins from natural sources is, right? So there are so many different pathways that are to be followed to get your protein in the desired form. So see, these are the different natural sources. There is wheat, there are peas, and these are all sources of protein, of course you know, right? And then what happens is these are all ground, and see, you are getting this dust, right? And from there can be wet extraction, there can be dry extraction. So you can have a look at all of these procedures if you're interested. But for our basically for our question answer session, there aren't many questions on these natural sources of protein purification and the pathways that are followed, right? So more will be discussed on the recombinantly produced proteins and how do we do that, and how do we finally purify proteins. So if you're interested in this is probably taken from a review paper. You can always go back and have a look at that. Okay, right. So we'll move on. So this is what happens when you want to produce proteins through recombinant DNA technology. Now what am I saying, this recombinant DNA technology term? What is it exactly? Right. So here there is recombination. Recombination means your natural DNA is present and then you modify it and then you combine it with a different DNA, and that is where recombination is happening. So see, this is your gene of interest which is present in blue. So this gene of interest which is present in blue This is the gene that codes for the particular protein. This is inserted into a plasmid. I am sure you all know what a plasmid is. Plasmid is a carrier molecule which is then inserted into bacterial culture. Here we are taking the example of bacteria because it is very easy to do it in bacteria and very common. There can be mammalian cells, there can be animal cells, there can be plant cells, yeast cells, right, insect cells. There can be various different types of host cells. And then what happens? This is grown, right? So this is actually a culture. If you look at this, it's a huge amount of bacteria growing together. You have provided the growth media. Bacteria is eating that up and it is growing. And then you break the cells of the bacteria open. And then you take out all the proteins from there. The bacteria will also have its own proteins, and then you separate out all those unnecessary proteins and only you collect your protein of interest. That you are doing using this chromatography. See, this is a column. So using chromatographic techniques, you're basically separating out your desired protein, and then you go for protein structure and function analysis, right? So what is the procedure? So gene cloning, this entire chopping off and joining of DNA sequences, this is called gene cloning. So gene cloning involves the isolation and amplification of the desired gene, right? Amplification means polymerase chain reaction is involved here, and then the gene is then inserted into a plasmid. So this is your DNA vector, which is the plasmid, which is a small piece of circular DNA that can be easily replicated in the host organism. Okay, the host organism is then transformed. What is transformation? Transformation is basically insertion of this recombinant plasmid into the host organism. That is called transformation. Okay, so transformation is happening. This is done using electrophoresis or chemical methods. Chemical methods include use of calcium chloride and heat shock. I am sure you've read about this during your 11th and 12th in school, right? Okay, so this allows the host organism to express the protein. What is expression? Gene expression means that that particular gene, DNA is getting transcribed into RNA and RNA is getting translated into protein. So that is your expression. Then what happens? Then expression of the protein involves including the host organism to produce the protein, and this can be done by adding specific growth condition. So we will have a look at all of this very soon, right? By adding a chemical inducer. So here one small chemical molecule is added and the bacterial genetic makeup is modified in such a way that protein expression will start, protein overexpression will start as soon as you add this small chemical compound. We will see what it is. Okay, then the protein is harvested. What is harvesting? It's like harvesting of crops. You basically, what do is you remove all the media and you centrifuge the entire solution and all the cells are isolated and collected into the pellet, and then you add lysis buffer to lyse the cells, right? Then see, through centrifugation or filtration process, this harvesting is happening. Then the purification of the proteins is a vital steps in this process. It involves removing any unwanted contaminants and impurities, which can be accomplished through various techniques such as chromatography, precipitation and filtration. So we'll have a look at all of these. Okay, now let's have a look at what exactly happens during the expression and isolation of recombinant proteins. Now here is a small correction. Instead of expression, it would be technically more correct to say overexpression. Why is that so? Because all the proteins in the bacteria, they are getting expressed naturally. But then your protein of interest is getting overexpressed. That is, there is actually a viral promoter sequence placed just before this gene of interest, right? So since this is a viral promoter which is often taken from T7 patch, and what happens is this viral promoter, it is viral promoter, so its activity is 5 to 10 times faster than the bacterial promoter, and what happens, huge amount of this desired protein gets produced. So when you run SDS page, you will see that compared to the bacterial proteins, your DNA of interest, your gene of interest is going to be your protein of interest will be huge, right, compared to the normal proteins in the bacteria. So this is better to say that it is overexpressed rather than expression, right? So let's see what happens here. At first we see there is expression in E. coli, right? So what happens, this is the conical flask, and then we have inoculated. What is inoculation? Inoculation means addition of the bacteria into the growth media, into the culture media. That is your inoculation. So inoculate media with the transformed cell culture. What is transformed cell? The bacterial cell which is containing the recombinant plasmid. Recombinant plasmid is having your gene of interest. Okay, and then we induce protein expression. Induction again means you are adding that chemical entity so that protein overexpression begins. That is induction of the protein expression. Then what happens? Harvesting. Harvesting means all this media is there and you pour it into these small cylindrical vessels. Then what happens? There is centrifugation. So all the cells, bacterial cells, they come down and they are present as a pellet. But then all the media, it is all watery liquid and you throw the watery liquid. Then what you do is you add lysis buffer, you go for sonication. So in sonication there is ultrasonic waves, and using these ultrasonic waves you actually break open the bacteria and you take out all the protein from inside the bacteria. And then again there is centrifugation. So if the protein is soluble, it will go into the soluble fraction. If the protein is not soluble, it will go into the pellet. Then what you have to do is, to solubilize the protein, you can go for urea or you can go for guanidine hydrochloride, so that the protein is completely denatured. If it is denatured, it is going to go into the soluble fraction, right? So these are different ways in which you can get your protein to the soluble fraction. There are also other methods, for example, inclusion body wash and all that. So if your protein gets like insoluble, it goes into inclusion bodies and it remains with the bacterial pellet. So then you will have to wash it with a specific buffer and you will have to try to refold it. So all of that happens. Okay, so let's see what happens here. Then after that, after this is done, you take it for purification. So here they have gone for nickel NTA purification. Nickel NTA purification we will learn very soon in the upcoming slides. Okay, this is a affinity chromatography based purification technique. So here what happens is the recombinant protein is having repeats of histidine. It is called histidine tag. Okay, at its N terminal or C terminal, you can attach repeats of histidine and the column will have nickel. Now, histidine has affinity for nickel and so the protein is going to get stuck to the column. So, only your protein of interest remains stuck to the column, rest of all the bacterial proteins go as flow through and you throw away the flow through. Then what happens is, one second. Yeah. So after that, what happens? After that, you add buffer with more salt. Then what happens? And the salt is actually imidazole in this case. So imidazole, if you know, it is actually the side chain of histidine, right? So imidazole is added and this is free, and so it will obviously have more affinity for this nickel. So all the imidazole goes and binds to the nickel through coordinate bond formation, and then what happens is your protein is freed and it is eluted out. So this is what is happening in this cell lysis and purification of polyhistidine tagged recombinant protein. Now besides polyhistidine, there can be various other tags. There can be GST tag, there can be so many different kinds of tags. We will learn about the tags also. Where is histidine? Histidine is often at the N terminal of the protein or it is at the C terminal of the protein. Okay. So we will learn about that very soon. Okay, now this histidine sequence, it can be present in the vector also. If you are making an expression vector, sometimes they provide that polyhistidine sequence, they provide the start codon. Which place will it be? Okay, so let's say that this is your protein, this is a chain which is folded, right? So at your N terminal, if you're using PET28A vector, it is going to be at the N terminal of the protein. This polyhistidine tag is going to be there, and just after the polyhistidine tag, there is going to be a cleavage site, so that you can cut it with a particular restriction, not restriction, proteolytic enzyme, and you can remove the histidine tag. Then you get only the free protein. Okay, so histidine tag and then cleavage site, and then your protein begins. If you are using PET28B, it is going to be at the C terminal of the protein. Okay, let's continue. So then after that, what happens is, now the protein is in a particular buffer. So we want the protein to go into a different buffer so that its physiological conditions are maintained, right? So this is often phosphate buffer saline. So you put the protein into a dialysis bag. So if you remember dialysis from your class 11th, 12th, 10th, dialysis bag has small pores in it. The pores are of the size lesser than your protein molecule. So the protein molecule cannot diffuse out, but then the ions and the buffer components diffuse out, and other ions from the buffer that is present outside gets in, and so this is how buffer exchange is happening, plus the protein is getting more concentrated. Then what happens after dialysis? You will concentrate the protein, so you will spin the protein, and there is going to be a filter, and again the pores of the filter is going to be smaller compared to the size of your protein. This is usually, I think, uh, uh, 300 Dalton or 3000 Dalton, like that. That is probably the size. And then what happens? The protein is definitely bigger, and so what happens? Buffers keep on seeping down, and the protein keeps on getting concentrated. So this is where this is happening, right? After that, you measure the absorbance. You can go for size exclusion chromatography, you can go for any other technique, you can go for SDS page, and then you get one intact band at the desired molecular weight position. Then you confirm that yes, this is my protein of interest, and the process has been successful. Okay, if you have any doubts, please ask me. You can write in the chat box and I will answer your doubts. Okay, now we will have a look at the vectors. So there is a difference between cloning and expression vector. You might already know this, but I'll just tell you once more, just to make sure that it becomes easier during the MCQs. So in the cloning vector, you can insert the gene of interest in the vector and you can preserve it, right? In case of expression vector, there are going to be start codon, there is going to be stop codon, there is going to be ribosome binding site, and there is often the tag. For example, his tag, GST, all these tags are going to be there for easier protein purification. So if you want to get protein out of your gene of interest, you have to transfer this plasmid. You have to isolate it from the bacterial strain containing this, which is often DH5 alpha, very commonly used, and you have to re-transform it into a different strain, which is BL21, very commonly used. So, and in the BL21, you have to express the protein. Now, why is it DH5 alpha? Why is it BL21? Please have a look. You can always Google and you will get the answer. So there are certain properties, there are certain proteases that are present in one, absent in another, right? So please have a look at all of that. So why exactly is it BL21 and how is it important in protein expression? There is a slide very soon coming up for you. There we will try to understand that. It is given in vivid details. Okay. So this is all about cloning vectors and expression vectors. I'll just give a quick read. Okay, so cloning vector is used to acquire multiple copies of the foreign gene. Okay, and they have features like the origin of replication, which you can see here. ORI-V is here. So ORI-5, this is the origin of replication, which helps the plasmid to make copies. Then there is restriction site. which you will cut so that you can isolate the gene of interest and you can put it into the expression vector after this, right? And also there is a what? Antibiotic resistance gene, because you need to make sure that the bacteria actually keeps the plasmid.
Ma'am, screen is not visible. Hello, ma'am.
What is not visible?
Ma'am, screen is not visible.
Screen is not visible. Okay, can you refresh once?
Yes, ma'am.
Okay, I'll see. Okay, I'm also refreshing. Let's see. Is the screen visible now? Is the screen visible now?
Yes, ma'am.
It is visible, right? You tell me, okay, I will not realize if it's not visible. Please tell me. Okay, so we'll go to where we were. So we were looking at the cloning and expression vectors. Yes, Kavya has raised her hand. Tell me, what is it? If you want to say something, you can write in the chat box. You can tell me by unmuting yourself, whatever is comfortable for you. Cloning. Cloning what? You want me to explain this more? Cloning and expression vector. Yeah, we were looking at cloning and expression vectors. Okay. Right. So this is your cloning vector. Can you see? Is the of the desired protein visible? Right. Okay. So it is visible, right? Okay. Great. Great. So yeah, for your expression vector, most important thing is ribosome binding site, where ribosome will come and bind and translation will begin, and then also there will be different regulatory elements such as the promoters, enhancers. See here, if you look here, you will see bacterial promoter and operators, right? Gene encoding repressor that bind to the operator. I'm sure you remember operon concept. We will brush upon that too very soon, right? So that is all. Initiation sites and stop codon, start codon, termination sequence, everything has to be there for the protein to properly express. Okay. Plus there also has to be selectable marker. So what is this selectable marker? For example, you want your bacteria to keep this plasmid within itself. Correct? So for that you need to give the plasmid some kind of special quality, which is, let's say, this antibiotic resistance, and you're growing the bacteria in an antibiotic containing media so that the bacteria survives using the antibiotic resistance gene present in the plasmid, and then it will not throw away the plasmid, right? Okay. Now what about this suitable expression system? Now this is a very interesting concept. See, applications of recombinant proteins range across different disciplines such as therapeutics, vaccines, structural studies, industrial enzymes, there can be food biotechnology, there can be so many more. Right. Now, this target gene and the expression vector, all of this is transfected into the host cell. Now, what is the difference between transformation and transfection? Transfection is the term used for plant cell. It is used for mammalian cells. But then transformation is the term used only for bacterial cell. Baki, the concept is similar. Right? Then what happens? What are the different kinds of host cells that can be used? So one example is your HEC293 cell, which is human embryonic kidney cells. There can be CHO cells. These are very commonly used mammalian cells, right? So mammalian cells are important for proteins that are huge. They require post-translational modification, and they are very useful in studying different kinds of pathological conditions, etc. Right. So then comes in the insect cells. In insect cell also post-translational modification is feasible. Insect cells are simpler to maintain compared to mammalian cells, and partial glycosylation can be achieved. Now we come to E. coli. E. coli grows very fast. It is easier to maintain E. coli. It is cheaper. But the issue with E. coli is that it is not very efficient for very huge proteins, and then again inclusion body, if the protein is not very soluble, that becomes a little problematic. Plus post-translational modification is not going to be permitted. Then there also is yeast cells. In yeast cells you can go for some post-translational modifications, and it is relatively simpler to maintain yeast cells compared to your mammalian cells. Correct? Have a look at all of this. This is a very interesting concept. So make sure you understand all of this. Right. Now we will go into the steps of protein expression and purification. Quickly we will just brush off. So see, protein, you've understood gene cloning. I'm sure you've understood transformation also. And then there is cultivation and induction. So for induction, what I was talking about is a chemical entity. So this chemical entity is actually your IPTG. So this is the IPTG. If you look at, one second, I'll have a marker. It will be easier for you to follow. It's not there. Okay. Acha. Okay. No problem. We'll continue. So this IPTG is a chemical which looks very similar to allolactose. Now, if you remember the operon concept, lac operon in E. coli, then what happens is we have one more slide very soon that is going to come up, and there you will be able to understand this better. But just to give you a small brief idea of this operator is present. An operator is present just close to promoter. So what happens? A protein gets formed, and this is it's a repressor protein. It comes and sits on the operator region. Then what happens? The promoter region is blocked, and so RNA polymerase is not able to come and sit on the promoter and transcribe the protein. It's not able to transcribe the gene, and so protein is not getting produced. That is the entire concept of lac operon. But then what happens when you add lactose then in the media? Then what happens? This allolactose comes and binds to the operator region and it comes and binds to the operator repressor protein, and now the repressor protein is no more able to go and bind to the operator, and so your RNA polymerase comes and transcribes the downstream genes. Here what happens is we are using a non-metabolizable allolactose variant, which is IPTG, and when IPTG is added into the media, then gene expression begins. So your protein overexpression begins when IPTG is added into the media. Okay, there's one more very interesting thing, which is PMSF. So PMSF is a chemical which is uh, a serine protease inhibitor. So it is preventing the proteolytic enzymes from chopping off your protein of interest. So when you are storing the protein, you will have to add PMSF, you will have to add a mixture of these protease inhibitors. Okay, this is called protease inhibitor cocktail. So cocktail means it's actually a mixture of many enzymes here. Okay, right. And when you're storing the bacterial cells, you will have to add PMSF, you will have to add sodium azide to prevent bacterial growth. Okay, now we will look at something very interesting, and this is what exactly happens inside the bacteria when protein expression is happening. Okay, so look here, this is very, what do I say, very mingled up thing is actually the prokaryotic DNA. Okay, inside the DNA there is some kind of modification. This modification is actually some region of a virus, which is lambda DE3 phage. This has been inserted into the into the nucleic acid, into the genome of the bacteria. This is having this lac-operon, plus it is also having this T7 polymerase gene. This gene codes for T7 polymerase. Now this gene is under the control of lac-operon. So what happens when you are adding this IPTG, then this gene expression begins. When you are adding IPTG, T7 gene expression begins. Without IPTG, this lac repressor is going to go and sit here, and it is preventing RNA polymerase from transcribing this gene. Now what is this gene? This gene is actually producing T7 RNA polymerase, which is a viral RNA polymerase. Now this viral RNA polymerase is coming and sitting on the promoter that is present just before your gene of interest. Now your gene of interest is under the control of this particular promoter. So T7 RNA polymerase, when it comes and sits here, then what happens? It is able to transcribe and you're getting the protein. Now there is also one more slight twist here. There can be one more plasmid which is called P-lyse S. So here what happens is, since lac-operon is a leaky operon, so there can be small amount of gene expression even when the lac repressor is present. Then what happens? T7 RNA polymerase gets produced and it keeps on producing your protein of interest. But that is very detrimental for the bacteria. If constantly so much of protein gets produced, then what happens? It becomes toxic for the bacteria. It is going to die, right? So to prevent that, there is this inactivating T7 lysozyme. This protein gets produced by this particular vector and it goes and inactivates the T7 RNA polymerase. But when you're adding IPTG, T7 RNA polymerase is getting produced in huge amount, then this inactivating procedure does not help anymore, and there is so much of T7 RNA polymerase that it can go and help in your protein production. So this is all about what happens inside a bacteria when you are overexpressing your protein of interest. So this particular vector, which is PET, this is an expression vector. This is a bacterial cell. It's written as host cell, and all the D3 component is there in the E. coli genome. It is engineered into the genome of E. coli, right? Okay, now we will see what exactly happens. So for protein expression, you have to select the host organism. you have to select exactly which strain, that is which variety of the host organism you want. You have to go for codon optimization. What is codon optimization? Codon optimization means that there can be different number of T7 RNA, different number of your tRNA carrying different amino acids, and these tRNA are having these anticodon loops. Now, the number of a particular tRNA which is complementary to a specific codon varies in different organisms. So, you want to select the codon for which there are more number of tRNAs for a particular amino acid. This is called codon optimization, right? So you have to check the codon optimized sequence for a particular organism. It is going to tell you exactly for which codon there are more number of tRNAs. So you have to select the one which has most number of tRNAs, and then you can go for co-expression, fusion systems, mutation, etc. Everything. Then there is protein purification. There are characterization of the physiochemical and functional characteristics of the protein. You can go for structural characterization. Okay, all of this follows. Okay, now we will start with the methods of protein isolation and purification. So right after this, our question answers begin. And then we have more topics coming up. Okay, so try to concentrate. Now, this is a method of protein isolation by precipitation. So you understand that precipitation of the protein happens when the charge of the protein, net neutral charge of the protein is attained. So that means the protein reaches its PI or isoelectric point. Correct? So for a protein, if this is the curve for solubility. So initially in this region, there what happens is the protein can get precipitated because there is, see, there are very less number of ions. Here what happens, there is no precipitation, the protein is very well soluble because the ions are there and the protein molecules are interacting, so solute solvent interaction is optimized. Here what happens, there is so much of solvent solvent interaction that there is less number of solute solvent interactions, and so the protein gets precipitated. So this is called the precipitation line, right? And here is your salt concentration. So what exactly is salting out and what is salting in? So initially when you increase the salt concentration very slightly, it helps in protein solubilization. This is the middle zone, this one. See, at low concentration, usually when you're adding the salts, then the solubility of the charged macromolecules increases because the salt screens out the charge-charge interactions. So low salt prevents aggregation and therefore precipitation, right? Then comes in salting out. So when you add too much of salt, what happens is the solubility of the macromolecules gets lowered because the salt competes with the solvent needed to solvate the macromolecules. So all the salt is now interacting with the water and your protein is left behind. It is interacting with itself and there are a lot of hydrophobic interactions and aggregation happens and protein precipitation happens. Now you try to solve this question. So what does the question say? There is a series called the Hofmeister series which gives you a list of ions and a specific order of arrangement of these ions, and it tells you which ion is exactly good for your protein solubilization and which ion is good for protein precipitation. So you have to choose the correct order of ions for the salting out process. Try to attempt this question. Just after this, we have the explanation and there we will see the Hofmeister series. Okay. So which one do you think? Is it ammonia? Is it K plus, sodium, lithium, and also in which orders? This is a very interesting question, very important question. Very simple question. As soon as you look at the Hofmeister series, you will know the answer. But it is very important for you to remember this, because these kind of questions get asked in competitive exams, and that time it is very confusing to actually answer the correct one. Okay, we'll have a look at the answer since none of you answered. So let's see. This is the series, and as you progress from the left to the right, you see that what happens? Protein solubility increases. As you are moving towards the right, protein solubility increases, protein denaturation increases, and protein stability increases. Sorry, protein stability decreases, and this is how it happens. So protein hydrophobicity decreases. So the ones which help in protein precipitation are called cheotrops. The ones which help in protein solubilization, they are called osmotrops. Urea and guanidine hydrochloride, these are your cheotrops. Okay. Ammonium ion is helping in protein solubilization. Okay. Now we come to the next question. If you are not able to catch up, please you can unmute yourself and you can tell me. I'll explain again. If I'm going too fast or if I'm explaining things that you already know, you can always tell me. Okay. Right. You try to answer this question now. Protein separation techniques are often based on the following properties. One is solubility of the protein. Just now we looked at solubility of the protein, right? Protein precipitation was actually based on solubility of the protein. This, okay, you're saying it's C. Charge, okay, C is 1, 3 and 4. So 2 is viscosity of the protein, 3 is charge of the protein, 4 is specific binding affinity. So you guys already know. Very good. Okay, so we'll have a look. We'll have a look at the right answer. Yes, the right answer is C. That is correct. So protein separation techniques mostly depend upon the charge, mass, hydrophobicity, pI and differential binding properties of proteins. They usually do not rely on viscosity of the protein, because viscosity is directly related to the concentration of the protein, right? Okay. Now we'll go to the this table actually tells you about the different purification techniques and what properties of the proteins they are dependent on. So you can go through this and it is going to give you a better idea. We will look at every one of these techniques in the upcoming slides. Try to answer this question. Oh, screen is not visible once again. Okay, one second. Let me check. I'll stop presenting and I'll present again, okay? Okay, tell me if you're able to see. Are you able to see now? Screen is not visible. Okay. Can you see my screen? Okay, you can see. Okay, great. We'll continue then. Right. So we were here. Right. So question on protein isolation and purification. During a successful purification scheme, it may be expected that specific activity increases, specific activity decreases, number of proteins in the sample decreases. So, which of these combinations do you think is correct? To give you a small clue, I have included the definition of specific activity here. Excuse me. So, specific activity is your enzyme activity. divided by the amount of enzyme present. Okay. So if I am purifying this particular enzyme, what do you think happens? B. Okay, so you are saying it's B. Let's see what is the right answer. It is A. So why is that so? Because when you are purifying this enzyme, what happens? The amount of enzyme, the denominator actually increases, right? And the number of proteins in the sample that decreases because you're only getting the pure protein. So the number of molecules will be obviously lesser than the one that was present in the mixture where there were many number of proteins. Correct. So what happens is the goal of protein purification is actually that the number of molecules of the isolated substance must increase during purification and the number of other molecules or contaminants must progressively decrease. There is a net decrease in the number of molecules in the pure fractions. Okay. Specific activity of the protein must increase. So, this, and what is the specific activity? This is a measure of its catalytic efficiency and it is defined as an amount of the substrate that is converted into product per unit time per unit of enzyme, often expressed in units per milligram of protein. I hope that is clear. Okay. Please stop me if you are not able to understand. Tell me if you want anything to change. Now, since we are talking about enzyme activity, here I have listed out some of very important formulae. So, in our upcoming slides, in the upcoming lectures actually, we'll talk about enzyme activity again. But to understand this specific activity concept, it is recommended that you go through these. Okay. What is enzyme activity? What is specific activity? And what is molecular activity? What are the classical units? What are the international units? What is turnover number? A lot of, many of you might already know all of these. But these are the places from which you get maximum number of numerical aptitude test questions. Okay, so please make sure that you understand this portion very well. Okay, all right. So we'll just quickly go through this. So unit of your enzyme activity is micromole of substrate transformed per minute and that is given in unit. Similarly, it can be referred to as moles of the substrate per second and it can also be called, the international unit is kcal. And similarly for specific activity also the formula is given, for molecular activity it is given and the uses of these three are also given. So I would say this slide is very important for you. Here also this percentage yield, purity, fold and total activity, specific activity, all of these formulae are given. Please make sure that you understand them properly. Make sure that you practice questions based on these formulae so that you can understand the questions better. Okay, so you try to answer this one. This is a small question. An enzyme is initially purified from a crude extract. So in your exams they are not going to ask you questions exclusively from one particular chapter, one particular unit. There are going to be mixture of 2 or 3 units in one single question. So you can expect questions where they are going to ask you about purification techniques and enzyme activity together. So it's very important that you understand these. Okay, try to look at this question and tell me what happens here. An enzyme is initially purified from a crude extract and its activity is measured at each purification step. So which of the following observations would indicate that the enzyme's specific activity is increasing with purification? So the total protein amount in the preparation decreases while the total enzyme activity remains constant. The total enzyme activity increases but the total protein amount also increases. Specific activity That is, enzyme activity per unit of protein increases. So you are saying it's C. Okay. While the total enzyme activity remains constant or increases less proportionately compared to the protein amount. And also your specific activity decreases, total enzyme activity decreases less than the total protein amount. So if you remember the formula of specific activity, this shouldn't be difficult for you. Let's see the right answer. Yes, you guys are very correct. The right answer is C. Okay. And also there are two more formulae for you to remember. I know that's too much, but then these are the places from where you can expect many questions. Alright. So yes, very good. It is C. Right. So your specific activity is expected to increase and the total enzyme activity remains constant. So, or it increases less compared to the other one. Now we will look at the different methods of protein separation or purification. So this is very important for this course. You can expect questions from these chromatographic techniques in every kind of competitive exams. So I have tried to make the slides very informative and easy to understand. You can please tell me if you want any other modifications or changes. Okay, now let's try to have a look at what the slide says. So understanding ion exchange chromatography. So from the title itself, you're understanding that there is going to be some kind of charge-charge interaction and there is going to be some kind of exchange. Correct? Okay. So ion exchange chromatography, also known as IEX, is based on the reversible electrostatic interaction between charged biomolecules, which are called, okay, ma'am, from where we would get numericals to practice any source? Okay. Numericals, I would say you can search for the previous year's question papers. Okay. Whichever exam you're targeting, please search for the previous year's question papers. Solve the previous year's question papers of GATE, of CSIR net. Actually, GATE questions are relatively more difficult compared to CSIR net numerical aptitude questions. Then you can go for DBT, you can go for other all of these different exams that you appear for. Besides that, you can get MCQ questions like previous year's questions, and you can also get different MCQ question books. So I remember we used to go for Catalyst, and also there are so many, there are Pathfinder books and all. Where to get previous year's question papers, you can go on the website. You can go on the website of that particular exam and you can download maybe 5 or 10 years question papers, previous year's question papers. Besides that, you can actually go and join groups in Telegram. So Telegram is very useful for all of this. You will get so many different groups where they will keep sharing so many study materials, so many. Actually, it's not available anymore. Is that so? Okay. So if Telegram groups are not available, or which one are you saying? Previous year's questions are not available on the internet. Go to the website of that particular exam. Okay, there will be previous year's question papers. Try to download those, and also you can search online. You'll get many such question papers. You'll get good websites. I will share the websites. Okay, I'll share the websites in the PPT if that's helpful for you. Okay, in the reference sections, I'll separately add the different websites that you can refer to for the questions based on these topics, and you can take help from there. Okay. So when you get the PPT, you can go back to the reference section and you can search for the questions. Okay, that will be useful for you. Okay, so we'll continue with the ion exchange chromatography. So look at this round thing. This is your cation exchanger and anion exchanger. So in cation exchanger, the protein is positively charged. Anion exchanger, it is negatively charged. And when will the protein be positively or negatively charged? It actually depends on what pH you are keeping that protein in. So based on the pH of the protein, it is going to be either positively charged or negatively charged, and it is going to get exchanged with ions. Correct? So oppositely charged functional groups on the stationary phase matrix. So in the cation exchanger, the matrix is negatively charged and it is often CM cellulose, which is carboxymethyl cellulose, carboxy group. You will know that it is negatively charged, right? Okay, pH is 4 to 7. For the anion exchanger, the protein is anion exchanger, the protein is negatively charged and the resin is positively charged and the pH is around 7 to 10. Okay, so what happens here? Let's have a look. So initially this anion exchanger is here and the protein being negatively charged is going to come and bind to it. Under low salt concentration it is going to come and bind to it, and all the other proteins that are uncharged or they are positively charged, they are going to pass as flow through. Then what happens? Your protein of interest, you increase the salt concentration. If you increase salt concentration, what happens? There are Coulombic forces, right? So your epsilon relative permittivity, this changes. Because of that, what happens? The Coulombic forces are now less and then the protein elutes. So negatively charged proteins, they are eluting here, bound proteins. Positively charged proteins elute first and then the negatively charged proteins are getting eluted when it comes to anion exchanger. So this is how you purify the proteins based on their difference in charge properties. Now we come to affinity chromatography. Now proteins have this characteristic property of binding to very definite chemical entities. These can be antigen-antibody interaction, this can be enzyme-substrate interaction, or it can be substrate analog. There can be protein-ligand interaction, there can be receptor-ligand interaction, lectin and polysaccharide, that is carbohydrate and carbohydrate binding protein. These two can come and interact. There can be nucleic acid and protein interaction, complementary nucleotide interaction, right? So many different interactions are feasible in nature. Then there can be metal ion and protein interaction. So your polyhistidine tag and nickel NTA chromatography comes under this. So metal ion protein interaction. This is also called IMAC, which is immobilized metal affinity chromatography. Okay. Then there can be glutathione and glutathione S transferase, or GST and glutathione interaction between proteins. So these kind of interactions are actually used by protein engineers to easily isolate the recombinant proteins from the host cell, from bacteria. Right. So look at this nickel NTA. What is happening? There is this agarose bead. To the agarose bead, your nickel is attached via this nitriloacetic acid. This is a chemical entity. Now to this nickel, two imidazole groups of two histidine residues present in the protein bind at a time. There are 6, so that 3 different nickel atoms can be bound to, right? At one time 3 different nickel ions can be bound to. This is called immobilized metal affinity chromatography. So nickel is covalently coupled to agarose using NTA, which is a hexadentate ligand, right? And then what happens? The natural or recombinantly synthesized protein can come and bind. Now what if the bacterial protein is having histidine in repeats naturally? Then also it can go and bind to your nickel NT chromatography. So this is one disadvantage, right? Plus there can be also cobalt used in place of nickel. Okay. So FPLC, or fast protein liquid chromatography, is a technique which uses nickel NTA chromatography to purify histidine tagged proteins. And elution is either biospecific, that is with an analyte like competitor, which is imidazole in this case, or it can be non-specific, that is increasing the salt concentration or pH concentration. Okay, I hope that is clear. Now we come to size exclusion chromatography. So we have a few more slides. We have just 2 more slides and then we go into the questions. So what is this size exclusion chromatography? See, there are particles of 3 different sizes, and based on the size of the pores in beads. The different sized molecules elute at different time. So first, when these molecules, the huge ones that cannot enter into the pores, they are eluted first. They are the large molecules. Then comes your medium sized molecules which have entered the pore but then not too many pores, right? So they come in just after these. Then are the smallest of molecules which have entered way too many pores and they are eluting last. So based on the time of elution, you can get an estimate on the size of the different molecules. Now whether you're getting a single peak or you're getting two different peaks, that tells you about the homogeneous population of molecules that are present or whether there are heterogeneous population, right? So your partition coefficient or KAV gives the affinity of a compound for the stationary phase. Okay, so this is VE minus V naught divided by Vt minus V naught. So VE is the elution volume, V naught is the volume of the space between the beads. This is also called void volume, and Vi is the volume of the space within the beads, and T is the total volume. So this is a formula that you must remember because there are numericals on this as well. Okay. So this is useful in getting the molecular weight of a particular compound. You can know, you can even go for buffer exchange using SEC. Okay. Right. Now we go into HPLC. Now HPLC is a very useful technique, very sensitive, very useful, but then very expensive as well. So what happens in case of HPLC is that there is normal phase HPLC and there is reverse phase HPLC. Okay. So in the reverse phase is more useful because it is based on hydrophobic interactions between the non-polar analytes in relatively polar mobile phase and the non-polar stationary phase. Okay, so what happens is analytes with higher hydrophobicity will interact more strongly with the stationary phase and will take longer time to elute. The desired analyte is eluted by gradient elution, that is different increasing concentration of salt is there in the buffer and the composition of the mobile phase is changed during analysis, right? So applications is that separation of nucleic acid. This is very useful in separating your double-stranded RNA from single-stranded RNA after in vitro transcription, and this is a very sensitive technique useful in separating different proteins, different nucleic acids and many other different biomolecules. Okay, so this is polyacrylamide gel electrophoresis. Multi-protein complex can be studied using native page where you do not denature the proteins and the interactions are retained. But in case of SDS page, you denature the proteins and you add a particular detergent which is SDS, sodium dodecyl sulfate, and it makes all the proteins negatively charged and you can visualize them in the form of monomers on the gel. Okay. Now we get into the question answers. So you try to solve these now. You can answer in the chat box. So the question says, you know that the protein you want to purify from a natural source forms a multimer with multiple subunits giving a molecular weight in solution much bigger than visualized on an SDS page. There is only a small amount of target protein in the total protein sample. Which of the following is an appropriate purification strategy? AC refers to affinity chromatography. IEX, you know it is ion exchange, and then comes in ACC. So how are you going to do? So there are two different things that you need to remember. Firstly, they have stated multimer. If they have stated multimer with multiple subunits, there has to be ACC. ACC has to be there. Plus they have also said that it is very small amount is present. Now this is separation from a natural source. So from a natural source, you understand that there are so many different impurities. You have to go through more than one size exclusion, more than one chromatographic methods. So which one do you think would be useful in this case? Okay, let's look at the answer. The answer says that the molecular weight of the protein in solution suggests that ACC would be appropriate. However, the filtration properties of ACC matrices compared to the capture properties of IEX or AC matrices means that only small sample volumes can be loaded on ACC columns, typically 1% of the column volume. As your target protein is already quite dilute, an unreasonably large column would be necessary for the purification of a useful amount of protein, and therefore it would be useful. So ACC would again dilute it, and therefore capture using IEX would be useful. After that you can go for ACC. So since this is taken from a natural source, the affinity chromatographic matrices, they are usually used for recombinant proteins, and so this wouldn't be a good idea, and therefore you can go for okay. So you are saying A. Very good. Very good. Now we'll go for the next question. Now this is differential centrifugation. In differential centrifugation, what happens when you spin at increasing speeds? At different times, different components of the cells get pelleted down. First, what comes is the whole cell, the nuclei, cytoskeleton. Then, with higher speed comes mitochondria, chloroplast, and lysosome. This is actually called that with higher speed comes the smaller ones like microsomes, small vesicles. Finally, comes in your viruses and ribosomes. So, with depending upon the density, you can separate out different components of a cell with increasing speed of centrifugation. This is actually called differential centrifugation, right? So now you try to answer this question. This is based on differential centrifugation and density of different components. Try to solve. Which among the following statements is or are correct? Number 1 is soluble proteins have higher density and lower sedimentation coefficient. Nuclei have lower density and higher sedimentation coefficient. RNA have lower density and higher sedimentation coefficient. Mitochondria have higher density and lower sedimentation coefficient. Which one do you think would be the right option? Try answering. Okay. So, yes. Okay. Right. Nuclei, the right answer would be D. So, nuclei have higher density and lower, okay, lower sedimentation coefficient. So, I need to look at this answer and I'll confirm with you in the next class. I think there is something wrong with this answer. But to tell you what sedimentation coefficient is, this is actually the formula for sedimentation coefficient, and its unit is often Swedberg's unit, and it depends upon the centrifugal force, depends upon the particles being separated, on the density of the particles, etc. Okay, so this is how you have to understand. Okay, this is a simple question. Try to answer this. This is a pathway, right? So A, B, C and D, these can be either recombinant DNA plasmid, expression vector DNA ligase and cloned DNA. Okay, so you're saying it's B. That is, A is DNA to be cloned, B is expression vector, C is DNA ligase, and D is recombinant DNA plasmid. Very good. Yes, that is the correct answer. That is the right pathway. Very good. Right? So yes, B is the right answer. Now we go to the next question. Which of the following is an example of the prokaryotic expression vector? Is it yeast? Is it virus? Is it bacteria? Is it mammalian cell? Prokaryotic expression, bacteria. Okay, let's have a look. Yes, very correct. Now we come to IPTG. That is your chemical activator, which was making sure that protein expression begins. So IPTG is added into the bacterial culture media to induce the protein expression. Which of the following options is not true about IPTG? So 1 says that it is a molecular mimic of allolactose. 2 says this is a metabolic, it is a part of a metabolic pathway. Okay, so you are saying it's D. D, which is not true. Yes, yes, very good. Yes, correct. So A is not true, baki these two are correct, right? So see, this is the thing about IPTG induction. There is lag promoter and then RNA polymerase is not able to bind and transcribe the gene because repressor is bound to lag operator. Then what happens? Your IPTG comes and IPTG make sure that repressor is no more able to bind to lag operator, and so the T7 gene gets expressed, T7 RNA polymerase gets produced, and the gene of interest gets expressed. So that usually happens when OD or optical density of the cells is usually 0.6 to 0.8. Okay, and lactose and allolactose, these are what? Isoforms. Okay, we'll move on to the next question. In size exclusion chromatography, the column matrix and solvent are selected to dash adsorption and elution is done using dash buffer throughout the experiment. So, column matrix and solvent are selected to what? Reduce the adsorption, minimize or maximize adsorption, and elution is done using same buffer or different buffer throughout the experiment. Size exclusion chromatography, if you remember, it is used to separate proteins of different molecular weights. Which one do you think would be the right answer? Column matrix and solvent are selected to, okay, so you are saying it's B. Let's see. Yes, you're very correct. It is done through using the same buffer, right? It is used to minimize the adsorption, and elution is done using the same buffer throughout. Okay. Now we come to anion exchange chromatography, and so proteins can be eluted from the matrix using a lower or higher salt concentration that reduces or increases the affinity of the protein for the matrix. Now since this is elution, you know what the second dash should be so that the protein elutes out, right? And to ensure that the elution happens, what salt concentration do you need? We have discussed this. You should be able to answer. It's simple. What is elution? Elution means that your protein of interest is coming out, right? Yes. So you are saying it's B. Let's look at the right answer. Okay, it is C. So yeah, higher salt concentration is required so that the affinity between the protein and the matrix is reduced so that the protein can be eluted, right? Now we'll go to the next question. amongst the following is false about HPLC? Which is false about HPLC? The sample moves through a column containing stationary phase under high pressure. High pressure limits diffusion and increases interaction with the chromatography media. It gives very high resolution of components. It separates mixture into compounds according to their molecular weight. See, there is a very obvious, yes, okay, let's see. Yes, very good. So, HPLC based separation is based on the affinity for the stationary phase, and it is definitely not on the basis of the molecular weight. Okay, now we'll move to the next question. Which of the following statements is correct? In dialysis, if you remember dialysis, we studied in that figure where proteins were dialyzed after purification. So in dialysis, the molecules diffuse through the semi-permeable membrane if smaller than pore size. Dialysis is used to remove low molecular weight compounds. In ultrafiltration, molecules migrate through a semi-permeable membrane under pressure. Ultrafiltration can be used for crude size fractionation and buffer exchange. Try to solve this question. I can already see that one is wrong. So try to eliminate that and try to answer. Okay, let's see which is the right answer. Very good. The right answer is 1. Right? So 1 says that all of these 3 are correct. Okay. Now we move on to the next one. We are probably left with 2-3 more questions and then we'll be done with today's question answer session. Okay. See, which amongst the following is incorrect about gel filtration chromatography? Now, gel filtration chromatography is another name for your size exclusion chromatography. So, I told you about this formula. This is why formulae are so important. They just keep coming up, right? So, particle size determines the void volume. Vt is the total column volume. V naught is the volume occupied by the gel space including matrix. Okay. Partition coefficient KAV is inversely proportional to the log of molecular weight. Yes, this is a very interesting point. And partition coefficient KAV is equal to, formula is given where VE is the elution volume. Okay, so you are saying that it is A. Right, let's see which is the right one. It is C, which is incorrect. So, partition coefficient KAV is inversely proportional to the log of molecular weight. It is actually directly proportional to the log of molecular weight in the resolving range. Right, we'll go into the next question. I'll still confirm if this is the right answer. I don't have the image here right now, but then we can always confirm, okay, whether this is the right answer or not. So I'll confirm and I'll tell you in the next class. Okay, so the first step in 2D gel electrophoresis. So what happens in two-dimensional gel electrophoresis is that along one dimension there is your separation of the proteins based on their PI or isoelectric point. Now different pH is given and so the proteins will travel and they will stop at the point where their net charge becomes 0. So this is the separation along one dimension and then the proteins are again separated normally like SDS page based on their mass. Okay, so this is called 2D gel electrophoresis. Now see what happens here. This generates a series of protein bands by isoelectric focusing. This separation along the first dimension, that is based on uh, isoelectric point, this is called isoelectric focusing. Okay. In the second step, the strip of the gel is turned 90 degrees and placed on another gel containing SDS, and electric current is applied in the second step. So what happens? The individual bands become visualized by interacting with the specific antibodies in the second gel. Proteins with similar isoelectric point become further separated according to their molecular weights. I even gave you the right answer while explaining. So the individual bands become stained so that the isoelectric focus patterns can be visualized. Yeah. So you are saying that it is B. Okay. I'll explain to you why it is B. The proteins in the band separate more completely because the second electric current is oppositely polar to the first electric current. In the first, what happens is, no, this is definitely not the right answer. So D is not the right answer. C, the individual bands become stained so that the isoelectric focus pattern can be visualized. No, that is also not the right answer. B is the correct answer, and A will not be the correct answer because you are not adding any antibodies. Antibodies will be added only in Western blotting. I'm sure you remember Western blotting. No, okay, sorry, I did not teach you. Okay, we'll look into Western blotting later. So, in this step, two-dimensional gel electrophoresis, proteins with similar isoelectric points become further separated according to their molecular weight. Okay, now we go into the last question for today, which is gel filtration chromatography separates proteins based on PI, morphology, secondary structure content, and effective size. Which one do you think would be the right answer? This is also simple. D. Yes, very good. That is the right answer. Okay. Thank you so much. These are the different references, and as you said that you want me to add the, yeah, okay. Sure. Please tell me.
Good evening, madam.
Yes.
Could you please explain the week 3 question 6 and 12?
Question 6 and 12.
Yes,
Which,
in week 3.
can you repeat?
Week 3,
Week 3.
in assignment.
Okay. Week 3. Acha.
Week 3, 6 and 12.
Acha. Okay. 1 second. Week 3, question 6 and 7. Okay. You please tell me if you can see the slides. I don't think you'll be able to see them now. Now you should be able to see. Can you see the week 3 slides?
Madam, I'm telling about it is about assignment, madam.
Acha, assignment. Assignment, this year's assignment,
Yes,
I am not supposed to discuss with you. I am supposed to discuss previous year's assignment.
but week 3 assignment already key also done, madam.
Okay. So the results are out. Okay. So
Yes.
I will write it for you. I don't have the assignment with me now. In the next class, we will
Okay.
okay, that's okay. You tell me which are the
Okay, madam.
questions 3 and 7, right?
6 and 12.
6 and 12, sorry. See, I already forgot. So,
Yes.
your assignment of
Already
3.
already that issue is in portal.
Okay.
In question and answer session, already that query was raised.
Right. If you have any other doubts, you can tell me. So in the next class, we will discuss these two questions, 6 and 12. You can also list out more questions if you want me to discuss them, because right now I'm not having the assignment. So, okay, in the next class, definitely. And also, you've given me one suggestion. Okay, can you also take question number 10 of week 3 assignment? Okay, so we are adding question number 10 to this list. All right. And in your this week's PPT, I will add the links of the resources from where you can practice MCQs and you can practice numericals. So that is one thing that I am doing. Okay. Any other suggestion, any other feedback that you have, you're always open to discuss. Tell me, if you don't have any other doubts, then we will stop the session today and we'll meet again next week. Okay. Thank you so much. Thank you for your time. This video will be uploaded on YouTube. Please go through it. Also, the slides will be provided to you. Thank you for being with me all through and listening. Thank you so much for your time and efforts. We'll end the session then. And you are always welcome to ask questions. Okay. Thank you so much. So we'll meet again next week.